Hello, I'm Linda Farmer. I'm Head of Food Research at the Agri-Food and Biosciences Institute in Belfast, Northern Ireland. And I have the pleasure of talking to you today about some work we've done about consumers of meat. So I'll just see if I can share the presentation with you. Okay. Right, so it's a very great pleasure to be invited to speak at this conference, the Science Powering the Future of Farming. And my only regret is that I'm not able to speak to you in person as was originally planned in March, but that I have to do it virtually, as it were, through this medium. So my presentation is on understanding consumer liking of meat. And what I'm planning to do is to talk to you about consumers, what we know, what are the gaps in what we know, and some of the work we've been doing to try and join the dots of the information that we have to try and gain a greater understanding. I'm going to talk a little bit about some examples of work we've done on beef and chicken, and then draw some conclusions. So first of all, consumers. Why do consumers matter? Well, the answer is basically that they've got the money, and all the money that supplies the entire supply chain comes from ultimately the consumers who eat the beef. And if they're not happy, then they're not going to want to pay money. It applies to beef, it applies to lamb, it applies to chicken, whatever meat we're talking about. So what do we know about consumer perceptions of meat? Well, there was a very nice study done by Maeve Henshin of Chagisk in Dublin, who reviewed all of the literature of work that's been done on what consumers like in beef. And she separated out the different characteristics as search, experience, and credence, and I'll explain those. So the green ones on this list are credence. These are things that you have to take on trust from the packet. What was its provenance, its origin? What was the animal welfare like, the production system? Is it organic? And then there's the stuff you can actually read on the packet: the price, the brand, the visible fat you can see through the plastic, and the meat color. And then lastly, there's the things that go on experience that you have to remember from last time you had it: the flavor, the freshness, the tenderness. So the interesting thing here is that flavor and tenderness come fairly well up the scale, and flavor comes higher up the scale than tenderness, despite the fact these are things that people have to remember from last time they had it. There's a lot of work being done under the heading of Meat Standards Australia. The largest part of it is done in Australia. But there's a lot done also in Northern Ireland and many other countries, including New Zealand. And that has led to a lot of information. We can understand the factors, the animal and the meat factors that actually have an impact on the eating quality for consumers. And the graph that's shown there is just an example of the work we did in Northern Ireland that shows that the factors that are really important for consumer liking, and then the hashed areas are where interactions are really important. And it's important to remember that it's the interaction of these different factors with each other which often has a very large effect. But what we also found from that study is that, and this was brought out by a PhD student who was working on this jointly across Australia and Europe, and that was that of all the meat eaten, the beef eaten in Europe, 19% of the grilled sirloin, 25% of the grilled rump, 53% of the roast topside was rated as unsatisfactory by consumers. So in other words, a fair proportion of the meat served is not meeting the standards that consumers are looking for. And that's not good news, given the fact that they've got the money in their pockets. This is another study reported by Oliver et al. In 2006, looking at German, Spanish, and British consumers. And what they were looking at is which was more important, tenderness or flavor, for overall acceptability of beef. And in all cases, flavor was at least as important as tenderness. And we found similar results in various countries using the MSA methods as well. So you can see there that 5 countries found flavour more important than tenderness. 2 found them about the same. And Japan found tenderness more important in the early trials, but then in the later trials, it was about the same. So again, flavour is at least as important as tenderness for the satisfaction of grilled beef. It's even more for roast beef, and it makes no difference what level of cooking, whether it's rare, medium, or well done. So what else do we need to know about consumer perceptions of meat? Well, it would be good to know what attributes they like or dislike, and whether or not people all like the same things, and if not, why not? And also what we really want to do is to know how can we give them what they like, and that means understanding the meat science behind consumer liking. Can we predict what they will like when the meat is at an earlier stage in the process? So the research we've been doing has been trying to fill these gaps, and we've been working on beef, lamb, and chicken. But today I'm just going to talk about beef and chicken. So if we think about beef, what attributes do consumers like? Which flavour compounds are really important? And what precursors of those flavour compounds do you have to have in the raw meat to give you a desirable flavour in the cooked meat? So here we have a consumer and a plate of beef. And what he's assessing is the flavour, appearance and texture, but of course also the odour compounds and taste compounds. And those arise from the precursors that are present in the raw meat, as I mentioned a moment ago, through various mechanisms of formation. But that's just the flavour and the odour. Of course, there's also the texture and the juiciness, and that arises out of the collagen, the proteolysis, the sarcomeres, the fat content, and all of that comes from the production and processing factors of the meat. So if we understand what it is the consumer wants and how it arises, then we should be able to dictate the right production and processing factors to give us the desirable quality that we're looking for. So what we're aiming to do to try and explain consumer liking is to take meat science, sensory science, and chemistry and biochemistry and link all those together using something called preference mapping. So what's involved in this is that we take, whether it's beef, lamb or chicken, we take a range of production and processing methods to give a wide range of sensory qualities. And then we conduct lots of different analyses on the same meat. So that'll include the traditional meat quality measurements, pH, shear force, the consumer panels and socio-demographic questionnaires, so we have some information on those consumers, sensory profiling with trained panels, and a whole range of chemical measurements of sugars, amino acids, fatty acids, antioxidants, and of course spectroscopy as well. And then we use multivariate statistics or chemometric methods to join all that information together. So here we have what will be an external preference map for grilled beef sirloin. At the moment, what we're looking at is principal component analysis. And that's just the black words there are the attributes defined by our trained sensory panel to describe the beef. And what we can see is that on the left side of the first principal component, we've got juicy and tender and also sweet flavour. And on the right side, we've got chewy, stringy, rubbery, sour flavours and cardboard aftertaste. And then at the top, we've got fatty aftertaste, fatty flavour and open appearance. And at the bottom, we've got crumbly texture. Now, if we now add on the consumer panel data, we can see that tenderness and juiciness assessed by the consumer panel agrees very closely with that assessed by the trained panel. And also flavour liking and aroma liking link in very closely with sweet flavour as well. So we can see that liking is coming out very clearly on the left-hand side of that graph. What we can now do is to add on some other measurements. So you can see in blue there, we've got some meat quality measurements. We've got Warner-Bratzer shear force, and it's not surprising that that comes out opposite to tenderness. Ultimate pH, likewise, is coming out opposite to the highest quality. And then if you add on some of the fat information, we can see that total fat is coming out at the top of the graph, together with fatty flavor and fatty aftertaste. And then we can add on certain flavor precursors, such as sugars and ribonucleotides. So this is an awful lot of information. As I say, you can see that there is logic to it, however, that the fatty attributes are linked to total fat. The flavor attributes are linked in with some of the flavor precursors, glucose and ribose there particularly. And some of the sour and less favorable attributes are linked in with some of the omega-6 fatty acids and the chewy texture with a higher Warner-Bratstein shear force. But it's very difficult to actually understand everything that's going on from a graph that includes flavor, texture, and appearance, because there's so much information there. So what we've also done is to take out flavor information and look at that separately to see if we can understand and manage flavor, and can we identify marker compounds that might help us to measure flavor. So here we have the same data, but in this case we're just looking at flavor liking, the flavor attributes and aroma attributes in black, and also flavor liking and aroma liking in red. So we can see that the most liked samples are those up in the top left of the screen here. What I've now added on are a whole load of the aroma volatiles and also some flavor precursors. So the top left here, we have the total amino acids, glucose, ribose, the sugars, and they're linked with the products of the reaction between sugars and amino acids, the Maillard products, which are shown in brown. So we can see that there is a rational link between the amounts of precursors and the amounts of the products formed from them. Another example here, we can see that we've got linoleic acid here, which is 18,2-cis-9,12. And that's linked in with 4 different lipid oxidation products, shown in blue and green, all of which are known to be formed from the breakdown of linoleic acid. As indeed is cardboard aftertaste, though it doesn't belong to any of those 4 compounds, but to another compound also formed from linoleic acid. And that's one of the important things that comes out of this graph, that while it may not be possible to measure some of the compounds that are really important for flavor because they're present at such very low levels, the fact that they're behaving in groups means that other compounds that are easier to measure may form marker compounds that we can use to anticipate where the best flavor lies. So this is the way that a lot of our work has been going, to identify, and now we're using those marker compounds to try and understand how flavor is formed. So in terms of beef, what we can say is that the consumer flavor liking is definitely linked to sweet flavor and to the Maillard odor compounds and to the sugars and amino acids in the raw meat, and it's inversely related to sour and bitter flavor and cardboard aftertaste and lipid-derived volatiles. And that gives us the potential to measure marker compounds amongst those compounds. So are all consumers of beef the same? And if not, how do they differ? Well, here we have a study, and in case my picture is coming over this for you as well, this is a study looking at the sorry about the noise there. This is looking at the some grilled sirloin from 6 different treatments, which are shown in the top right, though it doesn't matter too much what they are. And we looked at them with 3 different groups of consumers: in Belfast, Northern Ireland, in Cork, in the Republic of Ireland, and in Reading, in GB. And what we can see here is that while the pattern of the 6 different treatments is the same for all 3, there is a slight difference in the scores, a significant difference in the scores, with the GB consumers from Reading scoring higher. And this also shows up in this, this graph, table here. And what we can see is that there's a significant difference between the 6 different treatments in terms of the consumer scores. And there's a significant difference due to region because Reading was scoring higher, but there is no significant difference due to region treatment interaction. And what that is telling us is that the people from all those different regions like the meat in the same order on average. So it was always the same ones coming out on top. So why do these people differ between Reading and Cork and Belfast? We've established it's not because they have different likes. And we also established separately it's not because they're using the line scale separately or that there's any socio-demographic differences, due to perhaps differences in gender or income or age group. It's nothing to do with that. There may be a link with the motivation for choosing beef or consumption habit. And I'll just touch on these to explain a little further. Now, motivation for choosing beef, this is about whether or not the importance of healthiness of the product and the source of the product to the consumers. And what we can see here is that the GB consumers had more people who thought it was not important or of little importance. And therefore, they thought these attributes were of less importance than those people from Northern Ireland and the Republic of Ireland. It's difficult, however, to see why that should make a difference to how they score. So my money would actually be on the second of these, their consumption habits. And again, similar graphs, and what we can see is that the Reading consumers ate a lot more rump and somewhat more topside than consumers in the Republic of Ireland and to some extent Northern Ireland. So in other words, they're eating more of those cuts which we've already established tend to have a rather high level of unsatisfactory scores. So it may be that their habitual eating of poorer quality cuts is actually meaning that when we serve them grilled sirloin, they think it's pretty good. So in terms of beef, consumers differ due to region, but not necessarily in their preferences, not due to socioeconomic factors, but mainly due to past experience and also due to individual likes and dislikes. Because while we can use consumers in a large group to work out what the averages are, all individuals are different and they do have individual likes and dislikes. And that's what we use cluster analysis to look at. We have used it for beef. I'm going to talk about it more for chicken in a few minutes. Tenderness and sweet flavour are important, as is juiciness. Flavour liking is associated with natural sugars, amino acids, and the aroma compounds that are formed from them. And this work has been replicated in a number of different studies on beef and indeed lamb. And there are possibilities for marker compounds. So moving on to chicken, what we're interested in here is which attributes do consumers like, do consumers differ, and are there chemical markers again for liking. So again, much as we saw before, we did a whole range of different analyses on 14 different sources of chickens. We did the sensory profiling on 14 sources, and then we selected down to take 7 of those sources for consumer panels, instrumental analyses, and spectroscopy. So here we have 13 of our 14 sources. We dropped one out because it was too different from all the rest. And we can see that the principal component analysis is explaining 79% of the variation overall. We can see that the principal component 1 is separating on colour between white appearance on the right and yellow appearance on the left. And in terms of tenderness, up on the top right there, and rubberiness bottom left. But also on the same graph, we have chicken flavour and intensity of flavour, top right, and salty, sour flavour, bottom left. So what we did is select out the 7 orange treatments here for presentation to the consumer panels. And What I'm going to show you next is the graph on the left is it's the same principal component analysis, but just conducted with those 7 treatments. And what you can see comparing with the miniature of the graph I've just shown you on the right is that the overall shape of the graph is coming out very, very similarly. Again, we've got white appearance on the right, yellow and creamy on the left. We've got tender at the top and rubbery and solid at the bottom. So now we can add in the instrumental analyses and indeed the consumer analyses. So here we now have added in the consumer scores. So top right, tenderness, juiciness, overall liking, flavor liking, and aroma liking. And those are linking in with the tenderness at the top of the graph and also the intensity of flavor, chicken flavor, also in the top right quartile. What's also showing on this graph is the green cluster groups. I'm going to come back to those in a little bit. Again, it's quite hard to separate out tenderness and texture, texture and flavor looking at this graph. So we've looked at those separately. So this is the external preference map just for flavor liking. And we've got intensity of flavor on the right, bitter, salty, metallic, and chicken crisp flavor on the left, boiled chicken flavor at the top, and herby flavor at the bottom. And if we then put the consumer scores on, we can see that the attributes that the consumers like are the buttery flavor, the corn flavor. But that's very much the average consumer, which is the red words there. If we break up those 240 consumers into cluster groups, we can see that we have 4 different cluster groups here, one of which is markedly different to all the rest. So the cluster group at the bottom left, with 45 people in it, is liking exactly the opposite of the other ones. What we can also plot on are the different analyses, and here we can see again amino acids are linked with flavor liking and overall liking, but surprisingly sugars and the streccharaldehydes, which are Maillard products, are coming out in a slightly different area this time, and that differs from other work that we've done. Doesn't always work perfectly. What we did find is that there's quite a bit of variation between individual birds within treatments. We tend to think of chicken as being very consistent in its eating quality, but in actual fact, there's quite a lot of variation within one different source. So again, we were looking for possible marker compounds here. And here we've done a regression analysis looking for which of these different compounds have the best correlation or explain best the different predicted variables, the sensory profiling characteristics. And basically, it didn't do very well. Although there's very highly significant relationships, there isn't enough variation explained to actually start predicting what the eating quality is going to be from the amounts of volatiles or precursors that we have. We would need a lot more chickens and I think a stronger relationship to get that. Again, we were looking at are all consumers the same? And here we have the consumer scores from consumers in GB, in this case Harper Adams, and Belfast in Northern Ireland. Harper Adams is in Shropshire in the middle of England, close to the Welsh border. And what we found here is that there was no significant difference between those locations. And indeed, if we show the same graph as before, neither was a very significant difference in what they liked or disliked between the locations. So there was no interaction. The only significant difference was due to the different sources of chickens. So if we look at the overall liking preference map for chicken, we can see that this is the one that I showed you a few minutes ago. Apologies, but yes, I showed you this a moment ago. What I want to look at now are the different cluster groups. And what we can see here is that those different cluster groups are coming out in slightly different areas, which are associated with the overall liking scores shown in red, but are a little bit different. And what we want to look at is what is actually the difference between those cluster groups. Why are they different? And what we found is that the socioeconomics and behavioural attributes that they scored didn't differ. There was one very small difference about how often they purchased half a chicken. But basically, there was no socioeconomic difference between these groups. The differences, therefore, are all to do with how they score the chicken. And the one I've crossed out there is cluster group 4, which is so small that there's only 7 people in it that you really can't consider it. But if you look at the other 3 cluster groups, you'll see that cluster group 3, they really like their chicken. They just scored everything high. Cluster group 1, on the other hand, tended to score everything low, about 40 out of 100, except for treatment 10, which they scored even lower. But so something about treatment 10 they really didn't like. On the other hand, cluster group 2 really liked treatment 10, and that one they scored most highly. So there's a little bit of differentiation going on between the treatments by cluster group 2 and to a lesser extent cluster group 1. And we classed these as the people who liked chicken, the people who scored low, and the people who liked white chicken, because the ones that were scored highly by cluster group 2 were all those that didn't have any corn-fed or yellow colour in their diet. If we do the same thing just for the flavour graph, again, we've got different cluster groups. And we can see there's the 3 main ones there. Cluster group 2 on flavour liking is so small with 17 people that we won't consider it any further. So in this case, we've got 2 that are broadly associated with the average, and those are the larger groups, but you've also got a small group who like exactly the opposite down in the bottom left-hand corner. So again, there was only one significant socioeconomic parameter, and this was to do with household occupation. Again, we can mainly ignore cluster group 2 in these graphs. And what we're seeing here is that cluster group 3, which had the most difference in terms of what they seem to like, also had a higher number of people in technical type jobs and a smaller number of people in professional type jobs. I'm not sure that that is very politically correct, but that's the way the results came out. On the other hand, I think the most important differences here are the differences in what they like. And this was very highly significant. And again, your biggest group were chicken likers. They just scored higher for everything. And that is commonly the case with a lot of these consumer analyses. And then you have the next group who just scored low, but they particularly liked treatment 7. And then you had the alternative group who liked the opposite of what everybody else liked, who were very keen on treatments 3, 14, and 11. So what this is telling us is that there are subgroups within the population who like different things, and therefore there is the potential to market to those subgroups and identify the things that they're looking for to make sure that you're targeting the markets accurately. So most consumers like tender and succulent chicken, buttery corn flavors, flavor intensity, roast chicken flavor, and most consumers don't like rubbery and solid, bitter, sour, chicken crisp, herby and salt flavors. But there are subgroups that like different things, and that isn't linked to region or behavioral characteristics, but is due to inherent likes and dislikes and probably their past experience. So the other thing that we're looking at is, can we measure or predict consumer quality? And I'm going to touch on 3 different areas where we've done some work: Meat Standards Australia, compositional markers, and spectroscopy. So how can we predict what the consumer is going to like? Now, we've done a lot of work using MSA, working with Meat and Livestock Australia in the first instance, and then alone thereafter, and also with our own Department of Agriculture and the industry. And what that told us was that it worked very well for Northern Irish beef. And you can see here the residuals that show that there really wasn't very much difference between what was predicted for the quality that was predicted for a whole range of mussels and that which we actually obtained at the end of the day. And it worked even better if we amended the MSA system a bit to take into account specifics for Northern Ireland. So a little bit around the boundaries, because the consumers behaved a little bit differently. And some mussels they liked a little bit less or a little bit more. And indeed, we tend to have a certain amount of bull beef which wasn't available in Australia, and that needed to be added into the system. What we found, which was interesting, is that if you looked at how the predicted MQ4, which is the sort of score that underpins the MSA system, and compared that with the actual MQ4 score, MSA predicted very accurately for Australian consumers at the lower scores and for Northern Ireland consumers at the higher scores. And that's, if we look at the orange line here, these are the Australian consumers, and the green and blue lines here are all the Northern Ireland consumers. So for some reason, there was a difference in how the system was predicting the scores for Northern Ireland and Australian consumers. Ideally, what you're aiming for is to have all the lines running along the dotted line. In other words, where the two are exactly equal. If you look, on the other hand, at the ability to predict tenderness, that's quite interesting, because the MSA system was predicting tenderness extremely accurately for the Northern Ireland consumers, but less so for the Australian consumers. So obviously the Australian consumers were taking flavour into account a bit more, whereas in this very early work, the Northern Ireland consumers, it seems to have been mainly tenderness that was being predicted by MSA at that stage. And then we've already talked about marker compounds, and could those marker compounds be used to measure or indeed predict the flavour of meat? Well, it's something that we're looking at. We're not there yet because the relationships are quite complicated, and measurement methods for both flavour compounds and precursors in the raw meat tend to be rather slow. So it's still a possibility, but it's not one where we have any definitive answers. At the moment, we're using these techniques as a method for understanding flavour rather than as a prediction tool. We've also done a certain amount of work looking at spectroscopic measurement, and I know a lot of work's being done on this in New Zealand as well. And this is a graph done by my colleague Bruce Moss back in 2008, and it shows a very good relationship between the measured and predicted shear force using Raman spectroscopy. But what we've discovered is that there's an awful lot of questions you have to consider. Are you talking about calibration graph or a validation graph? I'm pretty sure the one on the right is a calibration graph. Would it have been just as good if we had used a different range of samples? And was the sample range wide enough and numerous enough in a wide range of studies to actually give us the potential to predict any sample that comes our way in the future? This is always a big problem working with the industry, to actually convince them that they need a very large sample set to actually get a predictive method going. What indeed are the spectroscopic methods measuring? Very often they are measuring compound groups rather than individual compounds, because the way that they measure in terms of the vibrations of chemical bonds and things of that nature tend to be not specific to individual compounds, but specific to groups of compounds. So we have to be quite careful what we think we're measuring here. And does it work for sensory or only instrumental tenderness? These methods tend to predict instrumental tenderness, Warner-Bratzer shear force, tenderometer measurements, but are less good for true measurements of tenderness using a sensory panel. So here's a bit of work we did on the chicken with hyperspectral imaging. And this is the instrument we were using. We were covering a range of 400 to 2,600 nanometers. And we had 16 chickens from each of 8 different treatments. So I think that's, oh, I can't do that sum quickly. It's 164 times 2. So that's 128 chickens that we had there. But what we found in terms of predicting texture and flavor attributes is that the amount of variation explained, shown in brackets there, is still only about 50-odd%, which is really not high enough to give us a predictive method. It was very highly significant, but not enough variation explained. We got very much better I'll come back to that point in a moment. We've done the same work in beef and lamb. We've got reasonably good prediction of instrumental measurements of texture, but again, not very good prediction of the sensory methods. If we look at some of the components of chicken instead, we got exceedingly good prediction of the fatty acids, ultimate pH, and sugars and their phosphates, and indeed T-bars, oxidation. But we need to be fairly careful that we know what we're measuring, and are we measuring groups of compounds rather than individual compounds, as I mentioned before. So in conclusion, what can consumer studies combined with other measurements tell us? In terms of prediction of consumer liking, we know that Meat Standards Australia works. The preference mapping has shown us that marker compounds help us with our understanding, especially with beef and lamb, a bit more complicated with chicken, and that hyperspectral imaging is good for some measurements, but not brilliant for consumer scores, better for some of the components that impact on the consumer quality. It tells us that there is a logic to the flavour of meat, that the flavour as perceived by the consumer is a natural result of the odour volatiles, the taste and the flavour release, which again arises from the flavour precursors that are present in the meat, the formation pathways, and indeed the underlying meat science in that meat. And if you look at beef in particular, what you can actually prove are the links between these various different aspects. And I'm not going to go through these individually, but we can see that all of these link together, and you can see the links between aging and proteolysis and glycolysis, the flavour precursors and the volatiles that are formed from them, for example. The third thing we can learn is that, and what the MSA folk have been telling us for years, is that consumers are exceedingly reliable. They are quite clear about what they like and don't like, and very consistent in terms of the attributes and treatments that are liked and disliked. But you do need large numbers of them in order to understand what's going on. But it would be a mistake to think that all these consumers are the same, and all these people have different likes and dislikes, which we know intuitively anyway. And these differences are not due to gender, age, region, or other socio-demographic factors. They are because people are different. It may be related to past experience, but it may also be related to the fact that we all differ in terms of our sense of taste, our sense of smell, and the way that our brains process this information. So, thank you for your attention, and I'd like to thank also my colleagues and students in Food Research Branch, many of whom are in this photograph, but not all. I'd also like to thank AgResearch and my colleagues there for the invitation to speak at this conference. And if you have any questions that you would like answers to, I have added my email address at the bottom there. I'm sorry not to be able to answer your questions in person, but if you wish to send me an email, I'll be delighted to hear from you. Thank you very much.